Is Abdul Elkadri from Medical College of Wisconsin, who will be speaking on intestinal organoid system to study rare pediatric intestinal disorders.
I could wing it.
Here it is.
I just need a timer. Yeah. All right. So this is not connected to that. I just have to.
Correct.
All right. Beautiful. All right. Good morning, everybody. My name is Abdul Elkadri. I am sandwiched between two powerhouses in motility, and I do not do motility. I came to the RASopathy world as a immunogastroenterologist, if that's a thing. I'm going to throw some crazy ideas out, just be a little spicy, because I think everybody brought up a little bit of something like we need some sort of a model to be able to figure things out. So there we go. So no disclosures. So I thought maybe we'll talk a bit about the overview of organoids, like what are they, some challenges and limitations, some success stories from other diseases, and some crazy possible applications. It's all kind of theoretical because this hasn't been really applied to intestinal issues within RASopathies, and then just some future directions. So intestinal organoids were created and developed by Hans Clevers in 2009, and it's basically a long-term culture that you can develop from biopsies or from an intestinal pluripotent stem cells, where it maintains the relationship between crypt and villi, which is something that you can't really do in single-cell models. It's basically easy, quote unquote. You need a couple of things. The ingredients that you put in the swill that you put around these organoids is Wnt to create crypts, so to signaling to create crypts, and then R-spondin 1 to allow for crypt hyperplasia, EGF for intestinal proliferation, noggin to induce crypt number expansion, and then to make this grow 3D, you grow it in this very fancy jello, this called Matrigel, very expensive jello, to allow it to avoid apoptosis, and in that it's bathed in laminin alpha-1 and alpha-2. That's what Matrigel has. And so this is just a photo of what it is. So that blob there on day one is an organoid. And in the center is the lumen. On the outside is the lining of the gut. And as you'll notice, there's a bunch of arrows there. Those are the crypts that bud outwards. And so you have crypt and villi within there. And so you basically have a mini gut system that you could do things to. That's a cross-sectional image of what's going on in there if you sliced it in half. So these can be created from multiple sources. During our workup in endoscopies, you can go in and grab an extra biopsy or two, which doesn't increase the risk, and then grow them in the lab. You can also take induced pluripotent stem cells from various sources and then create these through different mechanisms and ways. And more recently, there's been more development in the field in trying to create specific organoids, so esophageal ones, stomach ones, small intestine, and colon, so with their specific spice and kind of differences of what's going on. And what this whole system allows is, as you meet people with rare things, because again, we don't meet a ton of people that have these issues because they are rare, you can start to develop this library where you take these rare genetic changes and you can create a library that you can then go back to, because these can be kept almost indefinitely. And it's almost a perfect replication of what's going on within the patient, because you're not inducing the genetic change. You're taking the person's exact genetic change and you're keeping it the same. So there's additional things that you can do to these organoids. Here's a couple of them. You can do microinjection with a very, very tiny needle where you can inject things like bacteria. So that's been done in gastric organoids, where they put in H. pylori in there, and you can kind of see what the interplay is over there. Apparently, you can reverse the polarity and flip them inside out, and now you've got the outside inside and the inside out, and you can start to look at what the addition of molecules to that organoid does. You can grow them in a 2D plate where they're now a layer. And in that layer, you can start putting things on the top and seeing what happens with the gradient between the top and the bottom. And then the more interesting one is these microfluidic platforms. So you stick one of them right over there onto it, and then you can, using microfluidics, you can run stuff through it, you can run nutrients through it, and then see what changes. And these allow you to potentially run drugs and see what happens and what changes when it comes to the problem that you have within it. So challenges. This is not perfect. I painted a very rosy picture. These things are incredibly tenuous. They're epithelial derived, and so it's not really an organ as much as they're called organoids. It's not really an organ. Organs are made out of a lot of stuff. This is just one part of it. So people have started to co-culture multiple stem cell lines to create more complex systems. So as Dr. El-Shamas had said, you got some interplay between nerves and muscles and mucosa. Right, I'm only giving you one of them. So what they started to do is to mix them together by putting like an induced pluripotent stem cell from neural crest cells with gut to create a very air quotey enteric nervous system to try to determine what the interplay is between these two different cell lines. But it's very difficult to create and sustain. Easy said, right? The other bit is that these things, you have to sing to them, you have to play with them. But basically, the idea is that they're very tenuous, and there needs to be further optimization of the conditions because it's not really a great way to deliver nutrients to these organoids. They tend to kind of crump from time to time. So, what are some successes that we have? So cystic fibrosis was the first one. Cystic fibrosis is a problem with CFTR. It's a pump that moves chloride. And what Hans Klever's group or other groups had done is they had taken biopsies from patients with cystic fibrosis, and they've added forskolin. Forskolin is a molecule that causes an increase in cyclic AMP production, which activates CFTR. You activate CFTR, you start pumping chloride. Where chloride goes, water goes, and so you turn on a pump. And the organoids, because they're a circle, what happens is they swell. And you can see that in real time. You can put in the forskolin and suddenly they swell open and the bubbles become bigger. And you can do that in real time with a video or with photography, and you can measure what the changes are. So what happens in CFTR is that pump is not working. It's either not there or it's just broken and not working. And so they don't swell as well. So there was the development of the game-changing therapies such as ivacaftor and lumacaftor, where ivacaftor potentiates the function of CFTR and lumacaftor traffics it a little bit better. And when they added those in, what they are able to do is to show that it actually corrected the problem. And why it was important is because with ivacaftor, it really was only designed for about 5% of patients with cystic fibrosis, and lumacaftor was about 45%. 50% of patients had quote unquote rare genetic CFTR problems where they didn't really know if it was going to work. And they were able to prove that if you use the mixture and the combination of some of these drugs with this system, it actually worked and corrected the problem with very objective data with these swell assays. So it really allowed for a personalized assay because you can go in, take somebody's rare genetic diagnosis, put it in a little Jello dish. It's more complicated than that. And you can determine if the therapy works. Sounds cool. So another success story, I guess, is TTC7A. It's a very rare diagnosis with only a number of people who have been described in the world, where it causes this issue of multiple intestinal atresia. And these patients get these little strictures that develop within their gut that require resection, and they're incredibly unwell in infancy. And they have immune problems with severe combined immunodeficiency. And unfortunately, it's a typically fatal diagnosis, at least as of what we know of at this time. They're incredibly unwell, and with intestinal transplant and bone marrow transplant, we just don't know much about it. So there were two or three major groups in the world that had started to look at this disorder. A French group had taken the intestinal biopsies and tried to grow organoids. And what they found was they grew inside out, which is kind of weird. And so they had a phenotype. They had something that didn't make sense. And what they were able to do is look at what was going on within that system to try to determine why that was. And then they found that it had something to do with the Rho kinase pathway. So they got these really available Rho kinase inhibitors, so these ROCK inhibitors. And when they put it in there, it reversed that polarity. It flipped them inside out again. So they were able to say, hey, I think this actually fixes it. In reality, it actually didn't fully fix it. This is the photos that are there. So you'll see on the left, that's a normal organoid with the lumen over there. There's the budding that happens. The patient does not have the budding. Their lumen does not look the same. And when they added in Y, which is the ROCK inhibitor, you can kind of see that they have this bubble lumen situation going on. It does not have the budding. So it partially corrected it. You can better see it with the immunofluorescent, where the control is over here on the left and the patient is on the right. So if you'll notice, the alpha-6 integrin is in green. The pink is the apical portion of the cells with F-actin. And so pink on the inside, green on the outside, that's normal. As you can see, the patient, pink on the outside, green on the inside, it's flipped. You add in the ROCK inhibitor, it flips back again, and they added it also to the control. So here we are correcting it. It's not perfect, but it's at least an idea that's in theory. You're actually correcting something in the dish, and you're off to the races to try to determine what molecules you can use to try to correct this issue. And there are multi-center studies going on right now, because these are very rare diagnoses with people being born not necessarily right beside the world expert. And so they're trying to figure out something, crowdsource solutions. So what, right? Like, what does it have to do with RASopathies? The phenotype, oh, 5 minutes, yeah. The phenotype of the epithelial phenotype of RASopathies has not been fully described. And Dr. El-Shamas had kind of said that. We were looking for models to try to determine this, because in real life, this takes a long time to see this in patients, and it's trial and error. So from stuff that I've seen in interaction with patients with RASopathies, and how I got into this world is in seeing the CSFN families, I've noticed that they have a high amount of eosinophilic esophagitis, which kind of doesn't make sense to me. And I still don't understand why that is. And I'll talk a bit more about that. But the patients will sometimes appear to have occasional prolonged recovery post gastroenteritis with prolonged diarrhea. You say that's to families, and they all just kind of nod their head. It's not every single gastroenteritis, but there's times where I've gotten contacted by people saying, "Hey, I have a child who's in the hospital for 4 weeks now, and is this normal?" I've heard this before, and I don't understand why that is. And I actually got into this whole world when my patient who has a RASopathy developed inflammatory bowel disease, and it didn't respond to anything that I had. And it just didn't make any sense that two rare things could happen in somebody. So it had to have something to do with RASopathy. It just doesn't make any sense. The impact of that RASopathy cannot be ignored, and we do not know what's going on there. For example, the divergent colitis. That happens, but why is it happening in somebody with Noonan's? It's just a little odd. So some thoughts. This isn't something I'm saying that it's what should happen, but hey, let's take that EOE. We know I have this theory that patients with Costello syndrome may have an increased incidence of eosinophilic esophagitis. We know they have more atopic dermatitis. The mouse has atopic dermatitis. So we got something going on. Oh, look at that. There's ATROG, this RAS homolog that has something to do with EOE. So we have something of a vague, it's sitting in this area where maybe there's a pathway that's involved. There is eosinophilic esophagitis by name is this immune problem where the immune cells kind of infiltrate the lining of the gut. Maybe we can kind of look at what's going on the other side of the problem, on the lining of the esophagus, and figure out why they develop strictures, because up to 30% of adults with Costello syndrome develop strictures. So what has been done is there's a group at CHOP has created esophageal organoids, and they've added in interleukin-13 inhibitors. So this is Dupixent. And what it did was it stopped the differentiation and growth of organoids, indicating that the barrier integrity is better, blah, 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 blah. It basically stopped it from changing. And that's what happens. You change from an esophageal type phenotype to stricture phenotype. So it looks like it kind of stopped that. And again, they did another study where it led to improvement of remodeling markers. So what other things? Motility, that's the big issue, right? So with motility, you can potentially take biopsies or resection molecule or tissue from a smooth muscle, and you can create these spheroids, which is again an organoid made out of muscle. And interestingly, they were able to do this, and these things contract. So you leave them in the dish, and then you can kind of see this contraction over time by just looking at them in relaxation and contraction, and you get this rhythmic contraction that's over here. Well, now that you've got a model, you can put in something like a MEK inhibitor, and then you can see whether or not it changes it in there. I'm not saying that that's what you should do, because I don't think we know that that's what you should do, but I mean, I'd rather do this here than do it to a person and then have a tube up their butt to try to see what happens over time. Not that I'm saying there's anything wrong with that, but So future directions in motility, I think it may allow for a system to test the effect of therapies. We've talked about this. We want some objective data. You want to be able to show a graph and say, line go up, right? You want to be able to say this and point to it, instead of looking at things like the interplay between functional and neurological, which is incredibly difficult to separate apart. And I think, like, to do these studies, again, it's going to require repetition of very invasive motility studies, which families are willing to do for the risk there. But I think the idea is I'd rather do it in addition, get very controlled numbers. So the conclusion over here, I guess, to summarize, organoids, I think, provide a relatively easy way to create a patient's phenotype very specific to their genetic variation. So in rare cases where we don't see it all the time, there's a lot of challenges and limitations. I've oversimplified a whole ton of stuff. I fail all the time with these organoids, and I love them and hate them. But I think it may very well allow for a way to test therapies in a very non-invasive and very reproducible way. Thanks.
So questions? May I start off with one? So from a practical standpoint, this is something that can be very individualized to the patient. What is the practical time frame from getting a biopsy and growing it up and being able to throw a MEK inhibitor on it? Is this something that can be done in a week?
I'm smiling because in reality, it's a lot of work, but you have the lab running.
Yeah.
Somebody's there. You got everything ready. You got the jello ready. Everything's ready. You can go from a biopsy, and in 3 days, you get the blebs. 3 to 5 days later, you start having branching. You split them up, you passage them a couple times. Within about 2 weeks, you've got a bajillion of them. And you can throw these in a -80 and they're there forever. It sounds in theory very well when everything is ready. I think the problem that happens in research, and again, I'm like Dr. Olshamas, I'm a bit more on the clinical side. Got to get an IRB. You got to have a theory. You have to have an idea behind it. You have to have the funding. You have to have. It's just when you have everything together, it's so beautiful. It can take a week or two, and you have everything ready, and you have that person's genetic phenotype right there in front of you. In reality, science.
Well, I bet we've got a couple takers out here, right? So we'll find some collaborators. Other questions?
I wowed you. Sorry, out of left field again, 2 neuromotility powerhouses, and then I kind of throw this organoid thing in the middle. But I love that Dr. Olshamas had put the organoid there. Thank you. Shout out to you for the organoid. Thanks.
So has there been from other syndromes successful using a neuro organoid and a gut organoid and making them communicate? I mean, is this something that really practically can be done? Because just as what we were talking about, and we've heard there's so much complexity, you can't separate the neuro component from the muscular component, from the epithelial component. It's very complex.
I believe later today we have another organoid talk, and on the neuro side, there's also been some cardiac successes as well, where they interplay between them. So the gut, as always, is a little slow. That is a RASopathy joke.
Yes.
I'll die on that hill.
And the other thing is, like, where do you start? The gut is so complex because it's so long. You've got different sections are innervated differently.
I think you just get a biopsy, and then you kind of figure out where you're going with it. Again, my bias is immunogastroenterology, and again, I have this fixation that there has to be something on the other side of the equation, and the immune system is just not it. There has to be a RAS problem that has something to do with the fact that they have very odd immune problems that just happen to occasionally go into the gut. They do not always go into the gut. The neuromotility and motility problem they just dominate. So yeah, you could start from something. I think we just have to have some sort of a phenotype in a dish where you can throw something at it and see if there's a change without doing the mouse problem. This is literally the patient in a dish.
It's true. One final question. Any questions before we move on to the next? Thank you very much. It's
Thanks.
fascinating.